Imagine walking into a supermarket in the middle of January. Instead of the usual suspects like bland strawberries shipped from thousands of miles away, watery tomatoes, and rock-hard avocados, you are greeted by a vibrant array of fruits you have likely never seen before. There are golden, paper-husked berries that taste like a complex mix of pineapple and vanilla. There are grape-sized tomatoes that burst with an intensity of flavor lost to history. There are nutrient-dense African fruits, once rare, now piled high. These are not exotic imports brought in on airplanes. They were grown a few miles away, inside a vertical farm, on compact, bush-like plants that look nothing like their sprawling wild ancestors. This is the promise of the CRISPR Orchard, a new era of agriculture, where gene editing allows us to domesticate wild plants in years rather than millennia, turning nature's neglected orphan crops into the sustainable superfoods of the future. To understand the magnitude of this shift, we first need to look back at how we got here. For 10,000 years, humans have practiced agriculture the hard way. We call this the Neolithic waiting game. Our ancestors found wild plants with potential, planted their seeds, and simply waited. They waited for random mutations to occur that made the fruit slightly bigger, the taste slightly sweeter, or the plant slightly easier to harvest. They saved those seeds and waited again. This process, known as domestication, is what turned the toxic, spindly ancestor of maize into the corn cobs we know today, and the tiny, bitter berries of the nightshade family into plump, juicy tomatoes. But this process is agonizingly slow. It relies on chance, luck, and centuries of back-breaking labor. Today, with a global population hurtling toward 10 billion people, and a climate that is becoming increasingly hostile to traditional farming, we simply do not have centuries to wait. Enter CRISPR-Cas9, the molecular scalpel that has revolutionized biology. By allowing scientists to make precise, targeted cuts to a plant's DNA, CRISPR enables us to fast-track evolution. We can now look at the genetic blueprint of a wild plant, identify the specific genes that make it wild, such as sprawling vines, dropping fruit, or small size, and edit them to behave like a domesticated crop. This concept is known as de novo domestication. It allows us to take a plant from the wild to the supermarket shelf in a fraction of the time, potentially just a few generations. It is not just about tweaking existing crops to be slightly better. It is about unlocking an entirely new catalog of food. To understand exactly how scientists tame a wild plant, we must first understand a concept called domestication syndrome. When our ancestors domesticated crops, they unconsciously selected for a specific suite of traits. Regardless of the species, almost all useful crops share these characteristics. First is determinate growth. This means they stop growing at a certain height, remaining compact, rather than sprawling indefinitely across the field. Second is non-shattering. This means the plant holds on to its seeds or fruit even when ripe, rather than dropping them to the ground, which makes harvesting impossible. Third is gigantism, where the edible parts like fruit, seeds, or tubers become significantly larger than in the wild. And finally, reduced dormancy, meaning seeds germinate quickly and uniformly. In the past, achieving these traits required lucky mutations. Today, we know exactly which genes control them. The true power of CRISPR lies in multiplexing, which is the ability to edit multiple genes simultaneously. In a landmark breakthrough, researchers did not just tweak one trait. They targeted up to 6 different genes at once in wild species. For example, by targeting the SP gene, or self-pruning gene, scientists can turn a wild vining plant into a compact bush. By targeting the SP5G gene, they can alter the plant's sensitivity to daylight, allowing it to flower and fruit earlier in the season. By targeting the CLV1 gene, or clavata1, they can drastically increase the size of the fruit. In a single generation, a wild, unruly plant can be transformed into a compact, heavy-yielding crop ready for the modern farm. The theory is exciting, but the real-world results are already growing in laboratories and pilot farms. The most advanced candidates for these CRISPR orchards belong to the Solanaceae family, cousins of the tomato and potato. Let us look at the groundcherry, the poster child of de novo domestication. Native to the Americas, this small orange fruit is wrapped in a papery husk and has a unique, intoxicating flavor often described as a mix of pineapple, vanilla, and cherry tomato. In the wild, groundcherries are an agricultural nightmare. They grow as sprawling, tangling vines that take up huge amounts of space. Worse, they have a trait called abscission. As soon as the fruit is ripe, it drops to the ground. This forces farmers to harvest them from the dirt, raising food safety concerns and massive labor costs. A team led by Zachary Lipman at Cold Spring Harbor Laboratory and Joyce Van Eck at the Boyce-Thompson Institute used CRISPR to fix the groundcherry. They edited the SP gene to make the plant compact and bush-like. They targeted the CLV1 gene to boost fruit size by 24%. Crucially, they are working on the jointless gene to prevent the fruit from dropping, allowing for mechanical harvesting. The result is a plant that fits perfectly into high-density rows, holds its fruit for easy picking, and yields significantly more food per acre. Then there is the goldenberry, often called the new blueberry. It is a nutritional powerhouse packed with vitamins and antioxidants. However, like its cousin the ground cherry, it is wild and unwieldy. Recent work has focused on the erecta gene. By editing this gene, researchers created goldenberry plants with short, sturdy stems and a highly compact architecture. These dwarf goldenberries are perfect for urban vertical farms where ceiling height is at a premium. They produce just as much fruit as their giant wild relatives, but do so in a fraction of the space. We are also seeing the rise of the urban agriculture tomato. While tomatoes are already domesticated, their wild ancestor, Solanum pimpinellifolium, is incredibly resilient. It resists pests, diseases, and salt stress that often wipe out modern commercial tomatoes. Instead of trying to breed disease resistance into a modern tomato, which is genetically messy and difficult, scientists took the wild tomato and used CRISPR to give it domesticated traits. They created a tomato plant that looks more like a bouquet of grapes. It is extremely compact, matures in under 40 days, and produces fruit in tight bunches. This bunching trait makes it ideal for robotic harvesting in indoor farms. It retains the intense aromatic flavor and nutritional profile of the wild plant, but behaves like a polite industrial crop. This brings us to the infrastructure. The term CRISPR orchard is literal, but these new crops are not designed for the vast, open monocultures of the Midwest. They are custom-engineered for the controlled environment agriculture of the 21st century. Vertical farming has long struggled with profitability. Growing lettuce and herbs is fine, but you cannot feed the world on salad. To be economically viable, vertical farms need high-calorie, high-value fruit crops. But physics is a problem. You cannot grow a 6-foot tomato vine in a rack system with only 18 inches of clearance. CRISPR-edited compact crops solve this physics problem. A dwarfed goldenberry or ground cherry plant can be stacked 5 or 6 levels high. Recent trials showed that CRISPR-edited tomatoes for vertical farms reduced space occupation by 85%, while increasing effective yield by 180%. Furthermore, the predictable, compact shape of these plants makes them easy for vision systems and robotic arms to harvest, removing the massive labor cost that plagues traditional berry farming. One of the most radical frontiers in this space is heterotrophic growth. Startups and research initiatives are exploring gene-edited plants that can bypass photosynthesis entirely, feeding on acetate in the water rather than light. This would eliminate the single biggest cost of vertical farming, which is the electricity required for LED lights. While still in early stages, CRISPR is the key to unlocking these metabolic pathways. While trendy berries for Western supermarkets are the low-hanging fruit of this technology, the humanitarian potential is vast. We need to talk about African orphan crops. The African Orphan Crops Consortium is currently sequencing the genomes of 101 traditional African food crops. These are plants like baobab, marola, and spider plant. These crops are highly nutritious and adapted to local climates, but suffer from low yields and difficult harvest traits. Take the baobab, known as the tree of life. Its fruit has 10 times the antioxidant level of oranges, but the trees are massive and slow-growing. CRISPR could potentially create dwarf, early fruiting varieties that could be grown in orchards rather than harvested from the wild, providing a stable income for farmers and a reliable nutrient source for local communities. Similarly, gene editing is being used on staples like teff and millet to prevent lodging, which is when the plant falls over due to wind or heavy grain heads. A simple structural fix via gene editing could double yields without the need for new fertilizers. If the science is ready, why are these fruits not in every store yet? The barrier is a mix of law, economics, and culture. However, the regulatory landscape is shifting rapidly in favor of these crops, distinguishing them from traditional GMOs, which involve inserting foreign DNA like bacteria genes. In the United States, as of late 2024, the USDA-APHIS has significantly expanded its exemptions. Plants can now have up to 12 simultaneous genetic edits and still be exempt from GMO regulation, provided those edits could have theoretically occurred naturally. This is a game-changer for stacking traits like compactness, yield, and disease resistance in a single product. Across the Atlantic, the UK has implemented the Genetic Technology Precision Breeding Act of 2023. This act allows precision-bred organisms to be fast-tracked for commercial sale, positioning the UK as a testing ground for these new foods. Even the European Union, historically the strictest regulator, is moving toward a landmark compromise. A provisional agreement creates a Category 1 for new genomic techniques plants that are equivalent to conventional breeding. These would be exempt from the arduous GMO labeling and authorization process, though patentability remains a fierce debate. The economic case is equally compelling. The market for gene-edited crops is projected to surpass $19.9 billion by 2030. For farmers, the math is simple. Disease-resistant wild traits mean fewer pesticides. Compact architecture means higher yield per square meter, and unique superfruits like the ground cherry offer a premium product that competes with blueberries and raspberries, a multi-billion-dollar segment. Finally, there's the consumer factor. Will people eat it? The frankenfood stigma of the 90s is fading, replaced by a more nuanced view. Surveys in 2024 and 2025 show that consumers are largely open to gene-edited foods if the benefit is clear. They are skeptical of modifications that just help the farmer, like herbicide tolerance, but are enthusiastic about modifications that help them, such as higher nutrition and better taste, or help the planet by reducing food waste and chemical use. Successful commercialization will depend on transparency, marketing these not as engineered foods, but as restored or optimized versions of nature's best works, bringing the resilience of the wild back to our plates. We are standing at the threshold of a second green revolution. The first was built on chemicals and heavy machinery. This one is built on information and precision. CRISPR orchards represent a fundamental shift in our relationship with nature. Instead of bending the environment to fit our crops through irrigation, pesticides, and fertilizers, we can now bend our crops to fit the environment, whether that environment is a changing climate, a vertical farm in a skyscraper, or a smallholder plot in Africa. The wild superfruits are waiting. We no longer have to wait 1,000 years to invite them to the table.